And the first thing to say that this is a collaborative effort. So this has been a long-standing collaboration between several groups. So my group in Berkeley, Tom Terwilliger and Li Wei Hung in New Mexico. A recent addition, more recent addition is Matt Baker from University of Texas, Randy Reed's group in Cambridge, and then Jane and Dave Richardson at Duke University. So we collaborate to make Phenix what it is. And this has been mainly funded by NIH, but also industry as well, which we're very grateful to both places for sources for money to actually do the work. And then I'll return to this at the end, but there is a relatively recent paper that we recommend reading, of course, about Phenix. But that's also the thing to cite if you're going to cite Phenix in your publication. So when we started developing Phenix nearly 20 years ago, well, actually it was 20 years ago now, so it's some ways seems a long time, but some ways doesn't. We were really focused on developing a library, this computational crystallography toolbox that provides a lot of the algorithms for doing what we do, and then building Phenix on top of that. But over the years, this expanded to include a lot of other things. This toolbox is used by other software programs. And Phenix has some very nice interactions with a lot of different groups. This isn't all of them, actually, to bring in things like neutrons, molecular dynamics, force fields, some of the tools from Rosetta. And in the last few years, we've done a lot of work in the area of cryo-electron microscopy. Some of that with Wachu's group, but a lot of it with other people too. So you may be wondering, what is Phenix? When we started doing this, it was really, our goal was to try and to automate structure solution for crystallography a bit more than it was. It's really not possible to make macromolecular crystallography solution completely automatic yet. Maybe we'll get to that one day, but still not really possible. But we can make it a lot more automated than it was 20 years ago, for example. And this was important then because there was this new concept of structural genomics, solving a lot of structures to try and expand our knowledge of the relationship between sequence and structure. And the kind of goals that people had in terms of numbers of structures wasn't really going to work if it was all very manual. So this is one of the reasons we wanted to develop Phenix. We wanted to bring in modern programming concepts and develop new algorithms, of course, and have something that could be used by people who are new to the field, but also people who are very experienced and wanted to solve complex problems. So this is something that we built so we can develop this and maintain it in the long term. And hopefully some of you have seen that. You may wonder, why is it called Phenix? Well, it's called Phenix because of what's written here. That it's based around a programming language or a scripting language called Python, which 20 years ago was not a big thing. It was something that some people were interested in, but it had nowhere near the amount of exposure and use that it has these days. So Python is used everywhere, it seems. A lot of machine learning, but a lot of technology development in the software sphere. And so we wanted to develop this hierarchical environment for crystallography. So what are some of the key features of Phenix? Well, as I said, we use Python, and that's there because it makes it easy to script things, and it makes it easy for us to rapidly prototype and develop Phenix. It'd be a lot harder if we had to compile everything. And traditionally, we focused on crystallography, so there's a lot of algorithms for phasing, molecular replacement, model building, structure refinement, validation, etc. but you'll see that we've been able to extend those to expand into the area of cryo-electron microscopy. And we've been able to learn from some of the things we've done here, but also adapt to the specific needs of cryo-electron microscopy. And I think one of the things that we are proud of is that we rapidly develop the code and try to rapidly fix bugs and work with users like yourselves to solve problems. So in crystallography, these are the steps in crystallography, which some of you are probably familiar with. I'm not going to go through all of these, but just to say there's a lot of things that one needs to do to solve a crystal structure. And over the years, we've developed tools to deal with pretty much all of these things in crystallography. And as I said earlier on, automation was a big part of what we wanted to do here. And there are different ways that you can use Phenix. One is maybe less familiar to some of you, which is to use the command line. So on any of the machines that you're going to use, like a Mac or Windows, you can open up a terminal and type commands. And that's one way to use Phenix. And that's a perfectly fine way to do things. It's great. And there's actually a lot of automation going on here behind the scenes when you just type a command, figuring out what the data file type is, reading it and making sense of it, doing something useful with it. That's something that's done without you really having to worry about that so much. But I suspect that a lot more of you, if you've used Phenix, are used to using a graphical interface, the Phenix graphical interface. And if you've used it before, you're probably familiar with it. Just to quickly say, we can say more in the later sessions, but just to say that this is divided into sort of two parts. One on the left is projects that you've worked on. It's like a lot of software these days, there's the idea of a project or something that allows you to organize the information. And when you're working on a project, you can do all sorts of different things. Those are the things on the right. Those are the different tasks that you can execute. So these are programs, effectively. And there are tools for navigating through the things that you've run before, looking at them. And there's a few other things on top here. And we'll see in a minute, there's some links to some graphical software as well. This is a nice way to use the software because it can check things to make sure that you're putting the right things in the right boxes and give you feedback. That's the nice thing about a graphical interface. So I have to say, this is what I use most of the time. And we've been fortunate to work with the Qt developers and PyMol developers to have a nice integration between those things so that you can easily open your results, your structure, your maps, whatever, in Qt, for example, to do rebuilding. Very important when you're going through the process of refinement and validation, but also being able to look at things in PyMol to make figures and things like that. So there's a lot of things going on still in crystallography. It's a mature field, but there's still developments that are needed. And this, I'm not going to read through these, but just to highlight in the last few years, but there's a lot of things that have been developed in Phenix to help in the context of crystallography. That's not to say everything's solved. There are still challenges working at low resolution. These are the same kinds of challenges we have in cryo-EM, actually. Same challenges, again, across both techniques is how do you come up with a complete model, as complete as you'd like. And that can be challenging for multiple reasons. And refinement of models, certainly in crystallography, is not that automated. Generally, you have to make a lot of decisions. So this is where you need to be an expert. And there are things that we find challenging, like twinning in crystallography, and other things that make life difficult. And then finally, I just make this comment for both crystallography and for cryo-electron microscopy. ultimately what we're really looking at is chemistry. Typically, we're doing it to understand biology, but really it's chemistry. It's protein chemistry. It's the chemistry of small molecules. It's the interactions between the protein and small molecules or other things or other proteins or nucleic acid. And that's very difficult to automate. So that's something that really is one of the things that makes people, I think, good at structural biology, is having a good knowledge of chemistry. But I wanted to spend more of the time talking about electron microscopy, which I'm sure some of you are very familiar with. And this has been a technique that's been around for a long time, of course. And if we look back to the early 2000s, say 2005, the technique was in a place where it was possible to look at larger molecules. You know, these are ion channels. This is part of a virus. This is a large machine involved in protein folding. But they were relatively large, 250 kilodalton or larger, and the resolutions were resolutions that made it interesting to look at these things, but were not comparable to crystallography in terms of high resolution. So exciting and impactful, but not the same as crystallography by any means. Then a few years after that, Steve Luca and Wachu and others demonstrated that it was possible to get resolutions for reconstructions in cryo-EM that were good enough to interpret secondary structure and even build atomic models. These were reconstructions that's nominally around 4.5, 5, 6 angstrom. So that was an exciting thing to see, and it raised the idea that we could more routinely combine cryo-EM and crystallography to be useful with each other. And then a few years later, two groups actually published papers back-to-back in Science that were on this adenovirus at a reasonable resolution, resolution that it was possible to see things like side chains. And one was done by crystallography and one was done by cryo-EM. So it was fantastic to see cryo-EM maps that looked very similar to the crystallographic maps. And so this was a good demonstration that, given the right circumstances, you could get relatively high resolution, or resolution that could be interpreted de novo. But this was really a feature of having 60-fold icosahedral symmetry, very high symmetry in relatively rigid particles. Now, again, a few years after this, something quite important happened, and that was technology development in terms of detectors for electron microscopy. Existing detectors had used things like film and CCDs, what we had also used in crystallography in the past. But in the early 2010s, these new direct electron detectors were made, and they had a lot of very good things about them, but the very good thing was that they essentially were video detectors, they're video cameras. You could collect data at high speed, and that meant it was possible to correct the images for particle movement. This idea had been postulated by people previously, because if the sample moves during your exposure, you get a blurred image like this one here. But if you can spread this image out over several frames and then correct the frames, you can actually get an image that is much sharper, much clearer. And so that has been the big progress, I would say. One of the big progress things in the field has been this availability for direct electron detectors and much sharper, clearer pictures. And that has led in large part, along with other developments, it's not the only one, but in large part, I think has led to reconstructions that were very comparable to X-ray. This is around 2.8 angstrom resolution for a large assembly, a proteasome, but not long after being able to see proteins at something approaching, more typical crystallographic resolution around 2.2, for example, and being able to see small molecules, ligands, the kind of thing we would like to be able to do for drug development. And then more recently, really getting to atomic resolution. So in 2019 and 2020, these results at 1.5 and I think 1.2 angstrom resolution. So clearly we are seeing atoms here. And you can see things like hydrogens potentially. So cryo-EM has really progressed hugely in the last 10 years, approximately. It's a quite astounding success. So that's very encouraging, but it's also worth looking at, what kind of results are coming out currently. And I didn't redo this analysis for last year, but it will have changed, but probably not a huge amount is my guess. And this is just a graph to show the resolution of the cryo-EM reconstructions that we're seeing that are deposited into the database. And you'll see that the largest number is in this 3 to 4 angstrom range. So the majority of the cryo-EM work is still being done at sort of medium resolution, modest, I would say medium resolution, at the one end 3 angstrom, at the other end 4. 4 angstrom maps are not so easy to interpret. And there are still maps deposited at less than 4 angstrom resolution. And we are seeing an increase in the 2 angstrom to 2 to 3 angstrom range, which is great. And we can hope that increases. I'm sure if I do the statistics, we'll find there's more of these. But this is the area where most work is being done, is 3 to 4 angstrom. And if we compare that to crystallography, we would see that in that kind of range, that's about 7% of the structures that have been solved over time in crystallography. So we do solve, as crystallographers, people solve structures in that range, but it's not the typical range. So that does differentiate cryo-EM and crystallography a bit. It's going to change, I'm sure, and cryo-EM will catch up maybe to look a bit more like crystallography here, but it's probably going to take some time to get to that. But nevertheless, we face the same problems really in crystallography and cryo-EM at lower resolution. When you have nice high resolution maps, you can see what's going on. It's fairly obvious what's happening, and you can often see that even the kinds of atoms at high resolution. At low resolution, it's a lot harder. The maps don't really tell you what's going on. You might be able to see that this is where an alpha helix is, but figuring out what the side chain here is based on this map is going to be very hard. So that's a common problem. And it also turns out that if you look at the process of doing crystallography and cryo-EM, once you've got past the stage of collecting your experimental data and analyzing it in its first form, there's quite a similar set of things that need to be done, right? In both techniques, we have to look at the experimental data and assess how good it is. In crystallography, we've got this horrible thing called the phase problem to solve. So that's something that's a big part of crystallography. You don't have to do that in cryo-EM because you get the phases from the experiment. So that's great. But once we've got the maps, we're going to do things to improve the maps. And Tom will talk about that in the next presentation. And then we're going to try and build models or fit models into maps. And once we have models in maps, we want to be able to refine them and validate them. So these are all very common steps. So that means it's been possible to put together a set of tools in Phenix for cryo-EM relatively quickly. And so we are going to investigate some of these today and next week. We're not talk about every single thing, but we'll talk about many of them. And, you know, there's tools for analyzing maps, improving maps, segmenting maps, docking models, building models, refining models, and validating maps and maps compared to models. There are still challenges with cryo-EM, many, with automated model building. Things like the magnification of the map still aren't necessarily 100% correct. You know, how do we make the best map? There are been some good advances, which we'll hear about from Tom, but maybe there are even better ways that we can do things. You know, where do we have to look in the maps to interpret the map appropriately? Tom will talk about some of those things when we get to model building. And there is this issue that comes up more in cryo-EM. It's there in crystallography, but the resolution across cryo-EM reconstructions can vary quite considerably, and that presents some challenges. You know, and similar things apply in optimizing the models, that maybe that's low resolution. Sometimes these things are very large, and that's can be just a computational challenge. And sometimes the starting models are not so great. And there are still questions about how to really validate the results in cryo-EM. So we're going to talk about some of these advances, as I said, the rest of today and next week. If you want to get hold of Phenix, you can go to the web and download it. It's supported on, I think, most things you're likely to come across these days, doing work in your lab or at home on your laptop, maybe. There is a lot of documentation to read, so certainly encourage people to do that, but there are mechanisms to get help. We're very happy to help. We release a couple of official releases for Phenix twice a year. Typically, we have 2 official releases. But in between those official releases, we do put out what we call nightly builds. So we build a copy of a new version of Phenix every day. And if there's a reason for us to put that on the web, we will, so you can get hold of it. Maybe it will fix a bug that you told us about, or introduce a new feature that somebody was interested in. So certainly think about downloading nightly builds if you want to get the latest, greatest stuff. And it's great that you are participating in this workshop, but if you can't, or if you don't get everything you want out of this workshop, or you've got colleagues in your group that ask you, you should think about the Phenix tutorials. These are on YouTube, and so you can just search for Phenix tutorials and you'll get to this page. So there's a bunch of tutorials about lots of different things. There's about 30 of these, and to thank Dorothy for doing a lot of the work to get this going and up there. And there's things on crystallography and cryo-EM, so definitely worth checking out. And so I'm just going to finish up by saying thank you to all of the people that have worked and do work on Phenix. I mentioned the people who are part of the Phenix team here on the left, but we've had great collaborations with folks at other institutions like UCSF, University of Washington, Oak Ridge, a lot of other collaborators, and you know, to thank you, the users, for letting us know when things don't work, or whether there's something that you'd be interested in being able to do that you can't do with Phenix. We really appreciate the input. We certainly want to hear from you as much as you want to tell us what's going on. And then finally, again, just to thank NIH for their funding over the years, and the Industrial Consortium. So I'm going to finish. Oh, no, actually, one last thing, just to remind you, this publication in Acta Crist D it's 2019 now, but it's really worth reading because it covers some of the things that we're going to talk about today and next week. And a reminder to cite this paper. If you're going to cite Phenix in your paper, cite this paper because that's the most recent one.